Hello students, welcome to ICSE board class 10 biology episode 1.3.2. We are with unit 1 chapter 1 and 2. We have already completed some part of chapter 2, that is cell cycle, and in the cell cycle we have already seen about interphase and cell division. Cell division is required to be seen. So today in this session we are going to work out with cell division. So let us revise few things about the interphase. The cell requires to become mature, so it requires to undergo the process of growth and development. Once it becomes mature, growth and development occurs, it attains maturity, and once the maturity is there, then it enters into the process of cell division. So cell division, when the cell has reached certain level of growth through interphase, it enters the phase of cell division, where through a sequence of events the mature parent cell divides into two daughter cells. So from one cell, two daughter cells are going to form, and it is through the sequence of events. See, again the word sequence is there, that means it is a part of cell cycle. So we have already seen that cell cycle is equal to interphase plus cell division, and here we are talking about cell division. Interphase we have already seen in the previous episode. The phases are of various durations in a different cells, but the outline of sequence is going to remain same. So phases are going to be different for different cells. So for prokaryotes and eukaryotes is different, but the sequence, the outline is going to remain same. Now cell division consists of two major stages. Number one, karyokinesis. Karyo means nucleus and kinesis means movement. So division of nucleus is known as karyokinesis. And number two, cytokinesis. Cyto means related to cell or cytoplasm. Kinesis means movement. So it is a division of cytoplasm. So cell division is equal to karyokinesis and cytokinesis. Type of cell division. Cells in animals and plants divides into one of the two ways described below, depending upon the location of the cell. This we have already seen in the previous episode, that cell division occurs depending upon the site, depending upon the location of the cell. If the cell is somatic body cell, then it undergoes the process of mitosis. So somatic body cell means what? As for example, in animal, skin, tongue, alimentary canal, etc. are considered to be a body cell, or we can say somatic cell. Soma means body, so it is a somatic cell. So in this type of organs, or in this type of cells, you will see mitosis is taking place. In plants, as for example, leaf, stem and roots are the body cells where mitosis is seen. So sometimes they are asking one mark question, where do you see mitosis in animal cell or in animal, and where do you see mitosis in plant cell or in plants? So answer is, in animals it is seen in skin, tongue, alimentary canal, etc., while in plants it is seen in leaf, stem and roots. If the cells are related to the reproduction, that is germ cells, that is sperm and ova, or you can say androecium and gynoecium, then there is no mitosis, but there is another type of cell division which is known as meiosis. So there are two types of cell division, mitosis and meiosis, and it depends upon the location of the cell. So if the cells are located on the body or in the body, then it is undergoing the process of mitosis, and if it is in the reproductive organs like testes and ovary, then it is undergoing the process of meiosis, or if it is in the plants like androecium and gynoecium, then it is showing the process of meiosis. I hope you understood. Let us move on further how it takes place. So this is what it looks like, just zooming out. So this is how it looks like. Okay, yeah, this is the better one. So here if you see a single cell, mother cell, and here I have mentioned 2n. If it is mitosis, if the process is mitosis, if the process is mitosis, then it is going to form two daughter cells, and two daughter cells will be having same number of chromosomes. But if it is the process of meiosis, now where does the mitosis takes place? Mitosis occurs in a somatic body cell, in a soma means body, somatic body cell. If it is taking place in somatic body cell, then it is mitosis. It is what? It is mitosis. But if it is reproductive cell, if it is reproductive cell, then it will show the process of meiosis. So in mitosis the number of daughter cells are 2, 1 and 2. But if you see meiosis, then in meiosis the daughter cells are going to be 4. But one thing you have to be keep in mind, that in the process of meiosis half number of chromosomes are there. That means you are starting with 2n. You are starting with 2n. It reduces to n. So that means if a mother cell is having 100 chromosomes, then the daughter cells will be having 50. If the mother cell is having 80 chromosomes, then the daughter cells will be having half of it, that means 40. I hope you understood this one. This is what it means, the difference between the process of mitosis and meiosis. So mitosis is having diploid cell and the daughter cells are also diploid, that means 2n, while mother cells are having diploid cell and the daughters are haploid in the process of meiosis. So here it is diploid and here n is haploid. It is haploid. So this is a difference between the process of mitosis and meiosis. Another difference is that mitosis produces 2 daughter cells while meiosis produces 4 daughter cells. Where does one can find mitosis? In a somatic body cell. Where does one can find meiosis? In the reproductive germ cell. Here the number of chromosomes are same, here the number of chromosomes are reduced to half. So you can write all these things, then it becomes a definition. We will see that one afterwards. So this is how it looks like. Mitosis is simple, meiosis is complex, because in meiosis there are 2 phases, meiosis 1 and meiosis 2, while here there is only one phase, one complete. So this one is simple, while this one is complex. So let us move further. So now what is the meaning of diploid and haploid? The meaning of diploid and haploid I have mentioned here. A cell which has a complete set of chromosomes are considered to be a diploid cell. The total number of chromosomes in humans are 46, or you can say 23 pairs. That means in a human skin cell or any other cells except reproductive cells, the number of chromosomes in the nucleus is 46 or 23 pairs, or you can say 2n. So 2n represents the diploid cells, where n is the number of chromosomes, and it is due to the result of mitosis that we have seen in the previous slide. Here haploid cell, a cell which has a single set of chromosomes, are considered to be a haploid cell. In the reproductive germ cells, that is in sperm and ova, there are only 23 chromosomes, that is n. That means in the human skin there are 23 pairs is equal to 46 total chromosomes is equal to 2n, while in the reproductive cell it's half than that of skin cell. Hence in the reproductive germ cell there are only 23 chromosomes, or n. It is a result of meiosis. So in reproductive cell there are half number of chromosomes than that of a parent cell. So as I told, that if an organism is having 100 chromosomes in its body cell, then in its reproductive cell there are only 50 number of chromosomes will be there. So that is half. So there is a difference between the diploid and haploid. Diploid means complete set, haploid means single set, or you can say half the set. Here let us start with the process of mitosis. Mito means thread and osis means state. Cell division process was first described by Skeldon. This process takes place in the somatic body cells of organisms, as for example in animals skin, tongue, alimentary canal, and in plants it is leaf, stem and roots. The basic principle is the chromosome numbers are not changed, that means not altered in the process, it is just the duplication. It is a complex process where the mother and parent somatic cells divide to form 2 identical daughter cells. So it is complex, but if you see compared to the meiosis it is simple. Okay, mitosis is the type of cell. This is a definition. This one is the definition. This is the definition. So mitosis is the type of cell division occurring in a somatic body cells where 2 daughter cells are produced having same number of chromosomes as that of parent cell. So we have mixed all the 1 mark question here. Where is the process of mitosis occurring? The process of mitosis is occurring in somatic body cell. How many daughter cells are produced? 2 daughter cells. What is the characteristics of mitosis? The number of chromosomes are going to remain same as that of parent cell. An example, we have seen that in animal it takes place in skin, in animal it takes place in skin, tongue, alimentary canal, and in plants it takes place in leaf, stem and roots. Let us move to the phases of mitosis, or you can say phases of karyokinesis, that is mitosis. Mitosis is a part of karyokinesis. So it has 4 phases: prophase, metaphase, anaphase and telophase. So it is prophase, metaphase, anaphase and telophase. So here the simple you can remember like this: P M A T, prophase, metaphase, anaphase and telophase. So let us see the prophase part. What is the meaning of pro? Pro means first. It is the longest phase of mitosis. It is the first phase of mitosis and it is the longest phase. How much minutes it is? It is 50 minutes. It is the longest phase. It's a 1 mark question. This is 1 mark question. Which is the longest phase of mitosis? Prophase. If it is asked which is the longest phase of cell cycle, then the longest phase of cell cycle is interphase, because it is about 17 to 18 hours. 17 to 18 hours. But this is mitosis. So in the mitosis, the longest phase of mitosis is prophase. Okay. Nucleus is visible with a large and shortened, much like spring, and thickened chromosome. These are the characteristics. Now what happens in the prophase? We are talking about karyokinesis. That means we don't require any cell organelles. So cell organelles required to be removed from that. It dissolves in the cytoplasm. So cell organelle starts disappearing. First one, cell organelle starts disappearing. Then second one, nucleus and nuclear membrane start disappearing. Then okay, let me do like this: cell organelles start disappearing, nucleus and nuclear membrane start disappearing, centriole starts moving towards the opposite pole with the formation of ester rays, and finally forms spindle fibers. Chromatin network starts coiling and becomes condensed to form chromosomes with the visibility of centromere. And by the end of prophase, you get cell organelles completely disappear, nucleus completely disappears, centrioles reach towards the opposite pole with the complete formation of spindle fibers, but very few chromosomes are attached to it. Chromosome develops 2 sister chromatids, and in the plant cell, centrioles are not present, but still since spindle fiber network are formed with the help of polar caps. How does the prophase look like? The prophase looks like this. Here figure, if you see the figure, here figure A, this is figure A, this is figure B, and this is figure C. So let us talk about figure A. In the figure A, you will see each and everything. That means you are seeing a nucleus is there, chromosomes are there, nucleolus is there, all the cell organelles are there, as well as the most important part that is seen during the cell division is centriole. So centrioles are also seen here. So all these things are there. When the prophase begins, slowly, you see in B diagram, B figure, cell organelle starts disappearing. Chromatin, if you see here, this is chromatin, while this one is chromosomes, while this one is chromatids. So here chromatins are coiling to become a thick coiled chromosomes, and thick coiled chromosomes are splitting to form a duplicate arms of chromosomes, which are known as chromatids. And you see the nuclear membrane starts disappearing. Here you can see the prominent nuclear membrane. Here nuclear membrane slowly starts disappearing, and here you can see only the glimpse. You can see the blue line. This blue line is a nuclear membrane, number 2. So number 2 is the nuclear membrane. So here, so this is what the prophase looks like. The prophase, the cell is well developed. In the prophase, the cell is well developed, but as the prophase continues, slowly nucleolus, nuclear membrane, cell organelles starts disappearing. Centriole starts moving towards the opposite pole. On the other hand, the chromatin network starts coiling to become chromosomes with the visibility of centromere. You can see the red part here. This one, the red part is the centromere, number 6. So number 6 is the centromere. While at the end of the prophase, as the prophase starts fading, as the prophase starts completing, one can see only the glimpse of nuclear membrane. So one can see the glimpse of nuclear membrane. This number 2 is a nuclear membrane. You can see this blue dotted line. So this is the identification of prophase. While centrioles have already reached to the opposite poles, spindle fibers are seen, and some of them are connected to the chromatid centromere. Not all of them, right? So this is what the prophase looks like. Let us understand the where the attachment of spindle fiber takes place on the chromosomes. So here this whole part is a chromosome. This is a chromosome, while this one is a chromatid. So this one is chromatid, this one is chromatid, while the whole thing is chromosome. This one whole thing is a chromosome. So here on the chromosome, in the center, basically at the center, there is a point of attachment known as centromere. This centromere contains kinetochore, and on the kinetochore you can see the spindle fibers attaching. So what is kinetochore? Kinetochore is a protein. It is a site of attachment for spindle fibers located on centromere. And what is centromere? It is a part of chromosomes which helps in holding the chromatids. So let us move on to the second phase. Second phase is metaphase. It is of about 5 to 7 minutes. Here nuclear membrane is seen. You can see the nuclear membrane in prophase, right? You can see the nuclear membrane in prophase, but in metaphase nuclear membrane is completely disappeared. No nuclear membrane. You cannot see nuclear membrane. And one more thing, all the chromosomes are attached to the spindle fibers to the centromere kinetochore, and all of them are lying in the equatorial plane. This is an equatorial plane. It is the equatorial plane of a cell. All of them are lying. Now this is the best phase to study chromosome. Which is the best phase to study chromosome? Which is the best phase to study chromosome? Metaphase is the best phase. Metaphase is the best phase. Best phase to study chromosome. Why? Because all of them are aligned. All the chromosomes are aligned in the equatorial region. All of them are aligned in the equatorial region. So which is the best phase to study chromosome? This is the best phase to study chromosome. Metaphase is the best phase to study chromosome. Why? Because all the chromosomes are aligned at the equatorial plane, and all of them are attached to the kinetochore of the centromere that we have seen here. So here this brown part is a centromere, while the grayish part is the kinetochore, where fibers, these are the fibers which are attached. These fibers are the spindle fibers. They are attached to the kinetochore. It is completely attached and all of them are attached, seen in the metaphase stage. So let us move further down. Here anaphase. Now anaphase, it's a very active phase. Ana means back, pro means first, meta means middle. Pro means, we have seen here, pro means first, right? Meta means middle, or you can say after, and ana means back. Shortest phase, that is about 2 to 3 minutes. It's a very active phase. Main features are homologous daughter chromosomes, chromatids of metaphase now separate. They are separating from the middle, from the centromere. So from here they are going to separate. This is going to separate from here. One of the chromosome goes this side, another chromosome will go this side. So it is going to separate. It is seen in the anaphase stage. So here anaphase, homologous of daughter chromosome, two chromatids of metaphase now separate and being moved in the opposite direction towards the pole with the contraction of spindle fiber. So spindle fibers are pulling opposite direction. As a result, the chromatid splits and interzonal fibers are seen. During that time, various shapes of chromosomes are observed. That I will show, that will come with the later one. First of all, let us see how this takes place. So here, this is what it is. So here in the metaphase, chromosomes were at the equatorial region. In anaphase, they separate. This is moving this side, this is moving this side. That means due to the contraction of spindle fiber, the chromatid splits and move towards the opposite direction. So you can see that chromatids have split and they are moving towards the opposite direction. And late anaphase, the chromosomes late anaphase, the chromosome will reach towards the pole with the interzonal fibers. So this number 7 is the interzonal fibers. Number 7 is the interzonal fibers. These are the characteristics of an anaphase, as well as you can see the various shapes. This is L shape, this is V shape, this is V shape, this is V shape, like that. Various shapes of the chromosomes are observed. And how it looks like? It looks like this. So here, if you see here, this will give V shape. If it is pulled, then it becomes V. If it is pulled, this becomes L. If it is pulled, this becomes J. If it is pulled, this becomes I. So the various shapes are observed. Various shapes, V, L, J and I shapes are observed. Here V, L, J and I shapes are observed. Now how it is possible? Because of the presence of the centromere, position of the centromere. If the centromere is exactly in the center, if they exactly in the center, then it will be pulled and it gives V shape. So this is how it looks like. So here V shape, this one be L shape, this one be J shape, and this will be I shape. So here it is because of the presence of the centromere in the center. It is known as metacentric. If it is away from the center, see the center here, see the center here, this is the center. If it is away from the center, then it is metacentric and it is going to give L shape. One arm will be long and one arm will be short. Acrocentric. In acrocentric, the centromere is near the center, so one arm will be very long and this is very short. It is going to give J shape. It is near the end. It is near the end, not in the center, not away from the center, but it is near the end. It is known as acrocentric. While telocentric, it is at the end. So it is at the end. If it is at the end, it is going to represent as I shape chromosomes. So it is telocentric. So these are the various shapes of chromosomes that is observed in the anaphase. So if a person wants to study, if a student wants to study, or question is like this, that which phase is the best phase to study various shapes, then it is anaphase. So anaphase is the best phase to study chromosome. Which is the best phase to study chromosomes, shapes of chromosomes? Then it is anaphase. Just a chromosome, then it is metaphase. So this thing you have to keep in mind. I will, at the end of the session, I will give you the list of questions and we will discuss that part. Let us move on further. So this is how the anaphase looks like, and after anaphase, the last part is the telophase. So telophase is equal to end. Telophase is continuation with the cytokinesis. As soon as the chromatid comes together at the end of the spindle network, the telophase begins. So when they are moving towards the poles, towards the spindle network, if it is coming together to the centriole, then the telophase starts. It is the opposite phase of prophase. In prophase, everything was disappearing. Here it starts appearing. Spindle fiber starts detaching from the centromere, chromosome and the centriole starts disappearing. Chromosome starts uncoiling and forms a chromatin network. So here again it is going back to its original condition from where it started. It started from prophase, so it is going back to the prophase part. It is going back to the cell which was entering into the prophase part. In the prophase, when the cell was entering, at that time it was completely fully developed with all the cell organelles. Centrioles was appearing. Now here in centriole is going to disappear. Spindle fiber formation was taking place. Here in telophase, spindle fiber starts detaching and starts disappearing. The nucleolus was disappearing there in prophase. Here it is going to appear along with the nucleolus. So that's what it is. Nucleolus and nuclear membrane starts appearing. Cell organelles starts appearing. And if it is an animal cell, then you can say number 8, furrow will start taking place. Constriction will start taking place. But in the plant cell, cell plate formation occurs. Here I have shown only animal cell. So let us move to another diagram or the changes that is taking place in the telophase. Chromosomes reach opposite poles and spindle network disintegrates, but centriole remains, but slowly it also disappears by the time cytokinesis takes place. Chromosome uncoils, lengthens and becomes thinner, turning into the network or less chromatin threads. Nuclear membrane and nucleolus starts appearing. With the formation of nuclear membrane, nuclei and nuclear division, karyokinesis is over, and this is followed by the cytokinesis. So now when nucleolus starts appearing. That means karyokinesis is over and it is entering into the cytokinesis. On the other hand, slowly the cell organelles starts appearing. Cytokinesis means division of cytoplasm. In animal cell, at this point, the cytoplasm between the two daughter cells constrict. That is, in animal cell, a furrow starts forming and the plasma membrane at the middle. Because it's an animal cell, it does not have a cell wall, so it is very easy to constrict and it is very easy to form a furrow, so that the cell can divide into two daughter cells. Now this deepens, and that means it progresses towards inside. So it is from outside to inside, right? So in animal cell, the division starts from outside to inside, and it is known as centripetal cell division. While in plant cell, constriction is not observed. Why? Because there is a presence of cell wall, and cell wall is consisting of cellulose. Then how it is going to take place? It is going to take place with the help of cell plate, with the help of Golgi apparatus and endoplasmic reticulum. So in a plant, the division begins from inside to outside, and it is known as centrifugal cell division. Let us see how it takes place. The process of cytokinesis begins in animal cell from outside to inside, while in plant cell it begins from inside to outside. So I hope you understood this diagram. You can see here nucleolus is forming, 11 number is nucleolus, number 6 is uncoiling of the chromosome takes place, then number 5, that is the nuclear membrane appearing, centrioles and spindle fibers are detaching and they are disappearing, centrioles are disappearing. So this is what it looks like in the process of cytokinesis. So if in examination, if the figure comes in a figurative question, or if the figure is asked to draw, you have to draw this figure, label diagram. And if the figurative question is there, you have to identify whether it is a telophase or cytokinesis, because both of them are going to look alike. So how will you going to work in this one? You are going to work with this one, that you have to see the spindle fibers are disappearing, nucleolus and nuclear membrane is formed, cell organelle starts forming, and spindle fiber and interzonal fibers are disappearing. So these are the things. In the plant cell, you have to observe vacuoles, you have to see the formation of cell plate. That means it is a cytokinesis is taking place. Cytokinesis in plant cell, the cell plate formation will be little bit progressive. So this is how you identify. So this is the difference between animal mitosis and plant mitosis. Let us see plant mitosis. In plant mitosis, centrioles are absent. In animal mitosis, centrioles are present. Here no constriction or contractile ring is found, and here formation of constriction or contractile ring is found, but no cell plate formation. Third point, in telophase there is a formation of cell plate. Here telophase constriction is formed. Here nuclear and cell division is found in special regions like meristem. Here it is seen everywhere. Fifth point, it is from the division in plant cell is from inside to outside. It is centrifugal cell division. This is from outside to inside. It is a centripetal cell division. So you have to keep all these things in mind. What is the importance of mitosis? It is helping in the growth of tissues, organs, and thus organisms. So it is helping. It is helping in the repairing and damage of tissues and the organs. It is just like the need of cell division. It is helping in growth and development, repairing, regeneration, reproduction, right? As well as it also helps as a maintaining the number of chromosomes. So here it is acting as a asexual method by the lower organisms or lower animals to reproduce. Hereditary characters are maintained by replication of chromosomes. Characteristics of mitosis: it takes place in somatic body cell. It helps in tissue growth. It produces two similar equal daughter having same number of chromosomes as that of parent cells, or you can say it is just like the clone. So this is all about the mitosis. At this level, this information and this explanation to mitosis is enough, because it is related to 10th standard. So I hope you understood the whole process of mitosis. If you have any questions or doubt, let me know. We'll work it out some other time. Questions related to mitosis, I will put it in the next episode. I hope you understood this part. These are the notes, and 1 mark questions and differences I will give it to you later. Thanks very much, guys. I see you in the next episode.